Hi, I'm Connor Ryan, the Rhododendron Collections Manager here at Holden Forests and Gardens. Today we're at our David G. Leach Research Station. A lot of times people think that Holden only has two campuses, the Cleveland Botanical Garden in Cleveland and the Holden Arboretum in Kirtland, but we actually have a third campus, a satellite facility focused on rhododendron breeding and cultivation on the east side of Lake County in Madison. And so that's where we're here today to talk about rhododendron hybridizing. So we do rhododendron breeding at the Leach Station to make better landscape plants. Many people know rhododendrons can be kind of tough to grow, and so we're trying to make them easier to grow for people. And one of the main challenges in our area is the cold, and that's really what the focus of David Leach's work. When he was here, he actually is deceased and died in 1998, but he was very interested in adding a diversity of colors and forms into very cold hardy rhododendrons. And then David's successor, Steve Krebs, took that a step further and said, well, what's another thing that makes rhododendrons hard to grow? And that's this disease called root rot. And so he harnessed a species from Taiwan that's very resistant to this disease called root rot, and mixed these with David Leach's hybrids to create very cold hardy, very root rot resistant plants. And this is the species that Steve used to add root rot resistance. It's a species called Rhododendron hyperythrum. And this is a hybrid that is very cold hardy and very colorful. And today we're going to breed these together to hopefully create a hybrid that's cold hardy, colorful, and root rot resistant. So here we have a plant that's totally in full bloom. We've got these awesome white flowers. The buds are pink, they fade to white. And I wanted to introduce the parts of a flower so you can understand how we make a rhododendron pollination. So each individual flower makes up a group of flowers called an inflorescence. And so this would be one whole inflorescence, and here's another one, and here's another one. Rhododendron people like to call this inflorescence a truss. And so you can say we've got all these trusses all over the place. And then you have these individual flowers that make up the truss. And each individual flower is kind of like a flashback to biology in school. You have sepals, petals, and then you have the male reproductive parts and the female reproductive parts. And so rhododendrons in general have very reduced sepals. So you can't even see the sepals, but they're these little nubs at the base of this flower here. And then beyond that, you have this very showy white, which are the petals. Then on the inside of that, you have the stamens, which is where the pollen is contained. And then in the center of that, you have the pistil, which contains the stigma, where pollen would land and germinate and grow down the style into the ovary, where seeds will develop. So we have a flower that's been bagged and tagged. And the reason we put a bag over one of these flowers is that rhododendron flowers are pollinated by insects, and we don't want the insects to get into these flowers and make a accidental pollination that we weren't planning on. And so we totally cover it so that nothing can get in. And so I'll take this bag off. Usually we put the bag over a flower bud, so something like this, maybe cover it up before it even opens up. And so I'll take this tag off. Take the bag off, and then we have this flower. And another thing that I did ahead of time is I emasculated the flower. So I took off all of the pollen producing parts of the flower so that there's no chance that it'll self-pollinate itself either. And so on this flower you can see all of these anthers that are just full of pollen. I don't know if you can see it, but I'm just covering my hand in pollen. And there's a good chance that the plant will self-pollinate itself if you don't remove all of those anthers ahead of time. So I don't know if you can see, but I've taken all of those anthers off. And then the next step is pretty simple. I have a flower from a plant that I'm interested in breeding and crossing these two together. And so I'll just you can do this fresh, or you can store the pollen and do it later. But yeah, just take a flower. This is absolutely covered in pollen here. And so I'll just put it right on the stigma, which is this little green platform, basically. And I don't know if it's picking this up, but you can deposit pollen right on here. I would do it for every single flower. and just keep adding pollen. And the stigma itself is usually pretty sticky, so it doesn't take much to just, as soon as you touch pollen to the stigma, it just sticks right there, and you should be good to go. And then once you get every flower pollinated, we just put this bag right back on. You don't really have to be super gentle. And then I'll take my tag and write what the cross was. And you usually write the female parent first, so I'll write Rhododendron hyperrhythrum. And then I'll write the parent, which is a plant that we have in our greenhouse. Write the date, and then just tag it. And then we'll have this on this plant, so even if we are late to collect seed in the fall, the seed will still be kept in this bag, and we can always know what this cross was because we have the tag right on the bag and on the flower. We make these pollinations here at the David G. Leach Station, but we also make use of the collections at Holden Arboretum. So if you're ever at Holden and you notice pollination bags on the rhododendrons in the layer garden or the discovery garden, it's probably us.